Welcome back to this final part of the lecture, where we'll talk about functional magnetic resonance imaging. That's a method that will take a little bit of extra time to discuss here and introduce, because it's much more commonly used in neuroeconomics than any other method. In part because it reveals so much about the neural correlates of behaviors that we're interested in, and because it's relatively non-invasive and relatively cheap. So let's look at the methods in a little bit more detail. So MRI stands for magnetic resonance imaging. And what this means basically is that when you place a person in the scanner, like I am showing here on this slide, so the person is lying supine in the scanner here, then what happens is the magnet will do something to the hydrogen molecules within your body. And there are a lot of them because a lot of the body is made up of water, about 70-ish%, particularly when it comes to the brain. That number increases a little bit. And what you can see here is that these hydrogen molecules or atoms, they typically spin around in random directions when you're walking around in the real world. However, when you're placed in a strong magnet, these hydrogen molecules will align with the magnetic field. So some will align directly with the magnetic field, and the other ones will sort of have a opposite alignment. But all of them will be parallel to the magnetic field, basically. What this means is that there's no more random spins, but we can now control these spins. And we do this, or the orientation of the spins, by sending in a radio frequency pulse. So this is what the scanner then does. It sends in a radio frequency pulse. And this energy is absorbed by the spins of these hydrogen atoms. And so what will happen is they will be thrown out of their spin by absorbing this kind of energy. And this will lead to them sort of flipping their spin directions. And as soon as you stop sending in this radio frequent energy, they will then actually go back to the alignment with the strong magnetic field inside the scanner. And while they're sort of spinning back into the alignment with the magnetic field, they are in fact releasing energy. And this is energy that we can image. We can pick this energy up by measuring the radio frequent energy that's emitted by the hydrogen atoms. And this is something we can convert into a signal. So roughly speaking, this is how a scanner works. We have a strong magnetic field that aligns hydrogen atoms. We then send a radio frequency wave into the person, which will lead to these hydrogen atoms absorbing this radio frequent energy and being misaligned with the magnetic field. As soon as we turn off this radio frequent energy release, these hydrogen atoms will want to naturally realign with the magnetic field and release the energy they have absorbed. And this is something we can pick up via a radio frequency coil, and then later on process the images that we get from the scan. And the interesting thing there is that we can then make a nice image of the brain. I'll show you a couple of them in a second, because different brain structures actually have different hydrogen concentrations. And this allows us to image or visualize, for instance, the differences in gray matter versus white matter, right? So gray matter is the part of the brain that contains most neurons, whereas white matter is the part of the brain, is this kind of tissue that contains most of the axons and dendrites. And it also turns out that this is a highly versatile technology. So with MRI, we can do multiple things. We can use diffusion tensor imaging to visualize basically the direction of water movements. And that is related to the connectivity between white matter tracks. So you can see here in green, it goes from the back of the brain to the front of the brain, or vice versa. The blue goes from the top of the brain to the bottom of the brain, and the reddish colors go from the left to the right side of the brain. And those are the directions that neurons connect with each other, and also those are the fibers via which neurons communicate with each other. And there's some interesting correlational studies that look at the density of these fiber tracks and interesting behaviors that you can look at. Well, also, obviously, and that's what we'll talk about most, the MRI technology gives us access to BOLD fMRI. BOLD stands for the blood oxygen level dependent response. So changes in magnetization or in oxygenation of the blood across time that we're interested in. And that's what allows us to make these kinds of pictures with blobs on the brain, and also to sort of show activation differences across different conditions in our experiments. So, as another evidence of the versatility of this methodology, specifically of MRI, here are different images that the scanner can make of the brain. So we have T1 images, which are your typical anatomical images that we get and that you also see in papers published in neuroeconomics. So what T1 images show are a differences between white matter and gray matter, where white matter contain your neurons, and this is where we want to see activation, and gray matter contain the axons and dendrites, and we typically don't observe activations in those regions. What you see in black here is cerebrospinal fluid, which has multiple functions, one of them being that it protects your brain, so it allows your brain to sort of swim in this fluid, such that if you bang your head, you don't damage the brain immediately. There's also cerebrospinal fluid in this black area around the brain here. And then you can see that in this T2 image, which uses a different kind of sequence with different parameters, so different TRs and different TEs, you actually highlight the fluid-filled areas in the brain. And this is obviously relevant when you scan a patient and you want to have a clinical analysis of the brain. So you can then compare T1 and T2 images, and T2 images are often taken also to get additional information. And finally, we have T2 star images, which are very important for what we're interested in. They are actually the ones we use to capture the BOLD response. They are measured relatively fast compared to both T1 and T2, so we can scan the entire brain in a matter of 1 to 3.5 seconds. You get slices from the top of the brain to the bottom of the brain, which allows you to reconstruct a 3D image within about 3 seconds, maybe 2.5 on average in newer scanners. So this is what we'll be talking about when we talk about the BOLD response. And now moving on to the BOLD response, this is what we measure when we talk about functional MRI. And we are mostly interested there, or we are basically hijacking the fact that neurons require oxygen when they're firing, and the brain, what it does is it sends a larger amount of oxygen to regions that are active than actually can be consumed by these regions, which leads to an increase in the in the amount of oxygen or oxygenated hemoglobin within these regions. And this is something we can pick up as the BOLD response or as increased fMRI signal. So brain activation in the context of fMRI actually means more oxygen is being sent to a certain region within the brain that is active when neurons are active, and therefore when neurons are firing. But it's obviously heavily related to the anatomy of these blood vessels. And so regions, for instance, that do not contain blood vessels, which there aren't many obviously in the brain, cannot be imaged, but you can get some of these really strong activations where there are larger blood vessels, for instance. So it's somewhat dependent on the vascularization of the brain. As I mentioned earlier, we are going to use, or in fMRI, we're talking about T2 star images. They're also called EPI images, and I'm showing an example here. EPI stands for echoplanar imaging, which allow fast acquisition of these T2 star weighted images. Typical spatial resolution is about 2 millimeters in a standard 3T scanner, but you have to make a bit of a trade-off between how much brain you cover. So you might lose a little bit of brain when you have such small resolutions, and the speed of acquisition. So when you have larger resolutions, you might have a bit of a lower sampling speeds, which per slice vary between 50 and 100 milliseconds. And when I say per slice, what I mean by this is that the scanner, at least our scanner here, starts scanning from the bottom of the brain and then slowly acquires these slices up onto the top of the brain, and then starts again from the bottom of the brain. Within about 2.5 seconds, it covers about 35 to 38 slices to cover the entire brain. And each slice can then be reconstructed to get a 3D image every 2.5 seconds of the brain for the entire length of the experiment. There are some problems with these kinds of images. As you can see here, there's a big gap. So they are a bit sensitive to air pockets within the brain and your nasal cavities. There are one air pocket that lead to dropouts in, unfortunately, regions that we are very interested in, namely the orbitofrontal cortex down here. So you can see that in the T1 image, the OFC is here. You can clearly see the tissue here, but about at the same location there is a black area, and that's due to a dropout in this region. I'm highlighting this here again. But let's talk about briefly what makes these T2 star images functional. And this brings us to the bold contrast, which, spelling it out, is the blood oxygen level dependent contrast. And what I'm showing up here in the upper right hand corner is the metalloprotein hemoglobin. So this binds to oxygen. So when you breathe in, then oxygen is bound to hemoglobin, and this gets transported by blood throughout your body to support muscles, but also neurons with oxygen. And then obviously it gets used up and is pumped back to your lungs via the heart, and again in the lungs it gets bound again with oxygen. And one of the first demonstrations that the difference between oxygenated and deoxygenated hemoglobin leads to changes in MRI signal at a relatively fast time scale was by this study here, that these figures here, the pictures that I'm showing, by Ogawa et al. In 1990. And what you see here is a rat brain that is being scanned, but the air that the rat is breathing is heavily oxygenated. So it has a very high oxygen content. And you can see that when a rat that is breathing just normal air, you see these black lines everywhere throughout the brain. And these are basically arteries that are visualized. So in the context of, you can clearly see the arteries right here, right? Around the midline of the brain. So what this means is that when you have deoxygenated blood, then you have these kinds of signal dropouts within the brain, whereas when you have oxygenated blood, you do not have these, or you have much less of these signal dropouts within the brain. Actually, in fact, you almost see no arteries here. So that means oxygenated hemoglobin is diamagnetic or not magnetic. You do not have signal loss, as in this brain here, whereas deoxygenated hemoglobin is paramagnetic. So you observe signal loss, like in this image here. And the difference between oxygenated hemoglobin and deoxygenated hemoglobin is what we observe when we talk about the bold response. And this is the bold contrast, which is basically deoxygenated hemoglobin relative to oxygenated hemoglobin. And what you see is that, let's say we have some cognitive process that we're interested in, then we see that neural metabolism, synaptic activity goes up. This means that regional cerebral blood flow goes up, and this ratio here actually goes down. which means there's less signal loss or more increased signal, like shown here in this figure. So this is the canonical bolt response, which follows this kind of pattern all over the brain. So we have a sort of slow increase to a peak at about 5 seconds, and then it continues to decrease with a little bit of a dip back here. And we can pick the signal increase up because we have this compensatory increase of regional cerebral blood flow that actually supplies more oxygen to the regions in the brain that are active, which leads to this contrast here. So we have higher T2 star signal intensities that we observe. But it has to be noted that when we record these images, these functional images from the scanner, it takes a lot to pre-process these images. We have to go through a number of steps to clean them up, so to speak, before we actually can apply statistical analyses on these. And this is a sort of simplified pre-processing stream from, well, maybe the mid-2000s. Nowadays, this has become a little bit more complex, showing some of the initial pre-processing steps that we need to do before we get to do statistics and before we get to these kind of parametric maps that we're interested in, the blobs on the brain that you see in most of the papers. So let's go through each one of these step by step so that you have an idea of what it actually means to do an fMRI study and what these little, basically the summaries mean in the method section about what kinds of steps were applied to the imaging data. So first, we do motion correction. So one of the biggest problems that we have in fMRI research is the movement that participants go through. So they actually lie relatively still, but there is still some movement that we have to deal with. Even small head movements lead to major problems and can lead to either really large signal increases or really large signal drops, particularly at the edges of the brain. Here's the motion of a relatively good participant. You can see that so this is in millimeters. They moved about 2 to 3 millimeters in total in the X dimension. So that's from left to right. In the Y dimension, so that is from back to front, right? They moved almost not at all in this case. And in the Z direction, so that's from top to from bottom to top or top to bottom, they moved also less than 1 millimeter. And then you also have angles of rotation in different directions that you have to take account of. The good thing is that we can control for some of this if the movement is not too excessive, and a good cutoff is 1.5 to 2 times the voxel size that you're recording in, then we can actually take care of this. And we can actually use software to correct these errors, and then use the amount of error that was caused by movement as a regressor in our statistical models. The next problem we have is, now we want, before we go to looking at statistical images of all our subjects, we have quite different brains across subjects. So we can't just simply summarize activation here with activation in this relatively smaller brain with activation in this very differently shaped brain. So we have to bring them into one common space that allows us to perform group level statistics on these brains. And you can see that this is quite difficult with individual brains that have different sizes, shapes, and different types of foldings. This is a relatively large ventricle. This is a relatively small ventricle. So we have to bring them and squish them and fold them back into a common space. And we do this by using MNI space, or what we call standard space, which is shown here. So this is a Telerak atlas, and this is the MNI average template that's based on 152 brains. And there's again software that allows us to align this these different brains to one image. Now let's take a little bit of time looking at what our data actually looks like. So we are recording our data in voxels. A voxel is basically a three-dimensional like a cube, basically. But in the image language, you talk about voxels. Within the brain, you have about 150 to 200,000 in a typical brain that you're recording. And from each voxel, you're recording this time series, right? So this is one pixel in a sort of three-dimensional slice. That's what a voxel is. And each voxel is being recorded multiple times throughout a given experiment, right? So every 2 seconds, you get one image of the entire brain, and then you start again. And in this time, the subject is making their decisions about something that we're interested in, let's say whether to accept or reject a gamble in the scanner. So we get this time series of 3D brains, of 3D volumes of the brain that are basically recorded in slices. With each 2 seconds, a new volume is started. And we end up with a time series within each voxel that we can then analyze. And this brings us to the topic of model specification. So what is the next thing that we do with this time series here? We can specify a model, estimate parameters to test our hypotheses, and finally have group level statistics that we can represent in SPM. So let's talk about how we specify the model. This is basically showing the same time series that I showed you before, but now flipped sort of to the horizontal. So on the y-axis, we have the magnitude of the bolt response, and on the x-axis, we have the evolution of this response over time, so over our experiment. So let's say this is our entire experiment. What we want to see now is whether at specific time points that we as experimenters control in the experiment, we find that the bolt response rises and goes back to baseline in a way that is predictive, that predicts or correlates with basically behavior that we can sort of manipulate in our experiment. So the way we look at this is within an experiment where we know exactly at what time points we present certain tasks to the participant. And these tasks could be, like in this case, in this example, just the passive observation of faces. So at each arrow here, this participant is seeing a face passively. And let's say he or she is seeing this face for 1 second, and then the face basically disappears and there's a blank screen for a certain period of time, let's say 2 to 3 seconds. And then another face is shown for a second, and then another blank screen for 2 seconds, and so forth, right? So we know exactly what the participant is seeing or doing in the experiment because that is what we control as experimenters. But what we're interested in is we want to see whether this signal rise and wane, waxing and waning here, relates to something meaningful that happens in the experiment, such as viewing faces. And we can do this by building a model that is basically the optimal hemodynamic response if there was no noise in the system, and if the bolt response was perfectly following, viewing these faces in the context of the experiment. We do this by making these boxcars, which show that, first of all, participants are viewing the face for a certain period of time. The onsets of these boxcars are related to exactly the parameters of the experiment. So when is a participant seeing a face and for how long? That's reflected in this simple boxcar here. We also have information about exactly what the shape of the hemodynamic response is. So we take this hemodynamic response function and multiply these boxcars with the hemodynamic response function to get this predicted response here. That's what's shown in red here. We can then take this predicted response and look at, using statistics, how well it matches the observed response. So we get this sort of red line that is overlaid on the blue line, and you can kind of see that there is a perfect match or a very good match in this case here, with maybe some deviations on some points and some levels of noise. we can assess the extent to which this observed response matches the predicted response using statistics. And the statistic we used here is the general linear model. And that's what's shown here, basically. So in this case, y is our observed response. Again, remember, this is for each voxel, and we do this 100 or 200,000 times per brain. The x here are all the predicted responses based on our knowledge of the experiment, which we obviously designed as the experimenter, so we know at what time points what task is done by the participant, and also based on our knowledge of the hemodynamic response function. So the x here contains multiple predicted responses, such as the one in red shown here, and the y is then the observed response in a given voxel. The beta value is what we want to estimate, and our statistical software, such as SPM or AFNI, is going to estimate this for us. And then obviously there's some error term. So this is the statistical model that we use that basically gives us an indication via the beta value, via this regression parameter that is estimated here, of how much the observed response matches all of our predicted responses. And there can be multiple ones in here. So the more complex your experiment is, the more regressors we will have in this design matrix. The n here is basically the length of the experiment. So how many observations of the brain do we get per experiment? It's usually in the range of 500 to 1,000 different time points for an entire experiment. So this is then the statistical model that we measure, that we get to make these statistical parametric maps. So we've seen all of the different pre-processing steps. We've seen that we create a statistical model for each voxel, for each individual, which we then summarize in the end as a statistical parametric map across all of the subjects that took part in our experiment. So on average, across all of these subjects, how do these activations survive in, let's say, a t-test? Good. So that is how we run this. Another sort of note on the bolt signal: the signal is noisy, and it doesn't always follow this exact shape. It commonly looks like what we see here, where we cannot perfectly predict a rise, or we cannot perfectly see that the signal rises and falls with the predicted with the predictions with the predicted model, basically. But that's why we can counteract this using basically repetition, so signal averaging that is enabled via repetition. So we have to repeat each trial, each instance of an event, ideally about 40 times, 30 to 40 times. Finally, I want to mention that fMRI is not just about identifying where on the brain something happens, especially in neuroeconomics. We also use model-based fMRI nowadays. So that means we can use a model like the prediction error model here, or which we'll talk about next week in the lecture, or any kind of other model or value that we're interested in to make specific predictions about what shape or what magnitude at a given trial the bolt response should be in regions that actually compute or track specific values, specific parameters that we can manipulate in our experiment. And that is basically what this is showing. So we have parametric modulation based on, let's say, value, how much money a subject could win or lose on a given trial, and if there is a region in the brain that tracks loss, then it should specifically respond to variations in loss, but not to gains, and vice versa. Or if there's a region in the brain that tracks risk, then that should specifically respond to how risky a decision is on a given trial. So this is something that's very important because it allows us to have more mechanistic explanations about what a certain region actually is doing in the experiment or in the brain, whereas having just blobs on the brain doesn't really allow us to do this. This is how such a design matrix looks like, and this is how such a regressor might look like for prediction errors. So you can see kind of that with a regressor like this, you can no longer use your eye to make predictions about when when something rises or falls in the brain. It's a much more complex situation now. So I don't have time to talk about reverse inference here, but I want to encourage you to look this up on your own time. What are reverse inferences? I'm citing a paper here. Basically, it means that if I see activation on the brain, I cannot make an inference about what you are currently experiencing, what task you're currently doing, or what you're currently feeling. I can make an inference from my experiment to the correlation or the neural correlate within the brain while my participant is doing something that I control as the experimenter. But I cannot inference backwards from observing something in the brain to, well, in that case, this is the ventral striatum here, which is part of the reward system. So I might make the inference that, okay, the striatum lights up, or actually this is the amygdala, I believe. The amygdala lights up, so this person must be in a state of fear. Have a look at this paper at your own time, because it's quite an important point that I will make repeatedly in the lectures. As a final point, another tool that we have within imaging research is to use meta-analyses. This is also true, obviously, for behavioral research. We can use statistical methods that summarize results of multiple studies. And this is a very nice one from Bartra and colleagues that show the valuation system across over 206 studies here, showing that the ventral striatum is involved in valuation, but also the ventromedial prefrontal cortex. Something I'll also mention a lot in the lectures. And again, let me point you to Neurosynth, which allows you to do your own meta-analyses. So neurosynth.org. You can enter many terms and you will get such a meta-analysis that is a little bit more rough on the edges, but it actually is a very good approximation of these more effortful meta-analyses. Okay, I finish here. Thank you for your attention. If you have any questions, please email me, and I'm thankful to slides from these authors, Sonja van Welle, Gregory Burns, and Klaas Enno Stefan. Finally, here is a very good further reading that I'd recommend. The Glimcher, Kammerfair, and Poldrack book, Neuroeconomics, Decision-Making and the Brain, and there's a newer version out now. And have a look at Scholarpedia and the article on neuroeconomics. Okay, I'll see you very soon next week. Bye.